This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 527, recorded on December 28th, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here today in New York City, Dickson Despommier.
Hello, Vincent.
Welcome back.
Thank you.
This is our last episode.
It is.
Of 2018, and what a miserable day it is.
Well, are we going to talk about that?
You could look out the window. I
can't see anything. Cloudy. It's like a fog out there.
We have some kind of nor'easter, 13C.
We do.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
All right.
55 degrees and beautiful sunny skies. It's a gorgeous day.
Cool.
From Western Massachusetts, Alan Dove.
It is not a gorgeous day here. It's good to be on TWiV, though.
Welcome. And from Southeastern Michigan, Kathy Spindler.
Hi, everybody. It's a gorgeous day here. Blue sky. Way off in the distance, there are some clouds. 52 Fahrenheit, 11 Celsius.
And importantly for the Midwest right now, not a blizzard.
No. Right. That's not near us. Yeah.
So that's what came through here, this rainy stuff. Huh?
No, that's not blizzard either. We had the rain yesterday. That's different.
It's just not the same weather pattern that became rain? No? Okay.
No.
So that's what you told me last night, Dickson.
I thought it was.
Okay. I thought it was.
Alan,
you were almost here. You were almost going to be here.
I was gonna be down in the city, but family plans changed, and so.
Okay, that happens. But we could have had you in front of the wall of polio.
Yes.
In an effort to make ASM's podcasts the best that they can be, This Week in Virology wants to learn more about you, the listeners of TWiV. Please take 5 minutes to answer a short 12-question survey. They help us to collect data we need to seek sponsorship. Surveys at asm.org/TWiVpoll. That's asm.org/TWiVpoll. Thanks for your time and listening, subscribing, and helping us improve the show. Yesterday I tweeted this as per Kathy's suggestion on the last episode. And as usual, I went to Bitly and made a short link, and I said, I didn't need to do that. It's pretty short the way it is, right? asm.org.twivpoll, and it's memorable, whereas Bitly links are never memorable.
Well, and it's a good thing you tweeted it, because I wrote down last week, look at TWiVpoll, and I forgot. But then when I saw it on Twitter, I did. So it worked for me.
Good. And those of you who have taken the TWIM poll, asm.org/TWIM-poll, what a clever thing, right? You can take them both because it's slightly different audiences. Go ahead. It takes you a very short time. You'll feel a sense of achievement when you're done, like when you get anything done. Okay, you can check it off. Yep. Kathy, do you have any news about ASV?
Just a typical reminder for people to update their membership, pay your dues or become a member. And this is because the abstract deadline for the ASV meeting is coming up February 1st, as always. And the meeting itself is in Minneapolis at University of Minnesota, July 20th to 24th, 2019. And what I wanted to highlight for today is that the keynote speaker is Akiko Iwasaki. We mentioned that last time, I guess. Mm-hmm. And the plenary speakers are also available, and that information is all online now, both at the Minnesota meeting website and at the asv.org site. And from asv.org, you can easily get to the Minnesota site. So that's all I have to say about ASV.
I just went to pay my dues, and I noticed it's cool. You got a summary of our functions and services. Is that new? Mm-hmm. Is that new?
It's been up for probably almost a year.
It's new to me.
Well, you need to check out our webpage more often.
I haven't. Well, not to the dues page. You know.
Oh!
Haven't been there.
What you've done in the ASV in the past? Is that what you mean?
Yeah, it's on the dues page.
Yeah, that's always been part of your membership page, but maybe it wasn't visible in the old system. Yeah, so that's been more than a year.
It's cool.
Yep.
All right. Well, I'm there, but I need a credit card, so I can't do it right now. Otherwise, I would. You guys should add PayPal to the payments, because that could be done in a couple of clicks without adding anything. Just put it on your bucket list.
Yeah.
All right. More information about our AFM, acute flaccid myelitis crowdfunding efforts. We are at 56% of our goal. Actually, we are actually even better. Got a few donations this morning. Let's get an update here. Dickson gave $10,000. No. We are at $8,825, which is 58%. Thank you very much, all of you who have donated. And of course, if you donate even the smallest amount, you get a shout out. So let's shout out Kevin Waddell, Heather Barnica, Amy Barch, Linda Spatuzzi in honor of Bailey Sheehan, Tammy Ernst in honor of Braden Scott, Meredith Sanders in honor of Bailey Sheehan, Keith and Mikhail Sheehan in honor of our daughter Bailey Sheehan, Maria Oliver in honor of Bailey Sheehan.
Bailey's got a lot of support there.
Barb Jones in honor of granddaughter Bailey, Mike Park in honor of Matthew Park, Maya Sullivan in honor of Braden Scott, David Seletric, Tessa Park, Stephanie Langol in honor of Sam Langol, Stacy Becker in honor of Margot Becker, diagnosed September 2018, Jennifer Van Dyke Cesars in honor of Margot Becker, Bob Broilo, Motoko Bachman, David Perek, Matthew Garner, Deborah Bonagurio, Lenore Cummings, Catherine Meninger in honor of my friend Ann and her friend Margaret, whose niece is recovering from AFD. C. Brian Daniels, thanks to Vincent Racaniello for amazing me in each episode of TWiV and TWIM.
Nice.
Cool.
You can see a lot of these are relatives of someone with AFM.
They are.
Yeah.
By the way, I think that you should offer yourself as services for the graduation exercises on the downtown campus, because you read all those names and you didn't botch one of them.
I probably did, though. I probably did. How many names have to be read at the service?
Boy,
1,000?
a lot.
All of them.
That's right.
Kathy, can you take Sarah's email?
Sure. Sarah writes, Dear TWiV team, I was so bummed about the weather. Nary a mention. The weather from your various vantage points makes the podcast more vibrant, alive, real for me. I hope that other listeners who share my love of the weather will join in with gentle nudges to get it back into the show. Thanks for your hard work in putting on this entertaining and informative look at the world with its viruses. Sarah from Charlottesville, Virginia, where it is 8 degrees Celsius and partially cloudy at 1939, Sunday, December 23rd.
We have now unblinded our experiment.
Indeed.
Yes.
The fog is lifted.
Today we bring you another outbreak virus. To remind you, the American Society for Microbiology is supporting outbreak epidemics in a connected world on display at the Smithsonian National Museum of Natural History through May 2021. Here at TWiV, we're excited to see the topic of zoonotic viral diseases getting such a public platform. As a tie-in, we'll be discussing the latest info on one of the viruses featured in the exhibit. This month's segment is on hantavirus. If you're in Washington, D.C. area, be sure to go see the exhibit. It's free. And also check out the TWiV. What was that, 501?
Oh,
Okay.
Somewhere
That's no
in there.
problem. Microbe.tv/TWiV. And let's see. Oh, we are so far past 501.
501 is called Outbreak.
That's it.
Yes, that's it.
Gosh, it could be ages ago. So, hantavirus. Back in 1993, there was an epidemic of a highly lethal infectious disease in the Four Corners area of New Mexico. That's in the U.S. Otherwise, healthy individuals got flu-like symptoms, followed by pulmonary disorders, including massive accumulation of fluid in the lungs and death. Eventually, a new virus was identified from these individuals. It had been, relatives of this virus had been known to cause renal diseases. That's kidney Dickson in Europe and Asia.
Knew that.
Known to be associated with viral hemorrhagic fevers during the Korean War. And these are hantaviruses, commonly infect rodents, endemic in rodents all over the world. And this virus was isolated from the patients, and in rodents called the deer mouse, Paramiscus maniculatus, common in New Mexico. And it was called Sin Nombre virus. Originally, it was called Muerto Canyon virus because that's near where it was isolated. And the local resident says, no, we don't want our...
Well, and before that, it was even Four Corners virus.
Four Corners.
Right. And they didn't want it called that because that's a destination that they promote.
So, Sin Nombre, which reveals a aspect of the CDC that we didn't know about, that they actually have a sense of humor. Right?
Yes.
So this is an example of a zoonotic infection, which, of course, is the topic of the Smithsonian outbreak.
Well, an interesting side note here is that the epidemic was triggered by weather.
We're going to get to that.
Good.
We're going to get to that. And I know you love weather.
I do. I knew someone was involved in the epidemiology of this.
So what we think happened in the case of Sin Nombre, there was an increase in the deer mouse population,
right,
because there was a higher than normal rainfall,
that's right,
which caused a higher than normal yield of pinon nuts.
There you go.
Which both mice and humans like.
They do.
I like pinon nuts very much.
Yeah, they're good.
And the mouse populations increased, and therefore contact with humans, mostly getting into their homes and depositing feces and urine, which dries and aerosolizes as you inhale it. Now, in mice, these viruses are asymptomatic.
True.
Mice don't mind at all, but when you get dust on blankets or food storage areas, then you get infected.
But if
you're cleaning out your shed and there's rodent manure all over the place,
be careful.
Or you go to a motel and you see mouse pellets inside the sheets.
It's always a bad sign.
Always a bad sign.
You can always tell a mouse pellet,
but you can't tell them much. So this has another side to that, though, Vincent, because it wasn't just that there was a lot of pinon nuts one year. The next year there was almost none. But there were all these mice. Where are they going to go?
Ghostbusters.
So they went
where the food is.
They went looking for food in houses, which they almost never do. The same scenario in West Africa is the way Lassa fever is transmitted. Same way.
Mm-hmm.
Same scenario even.
So it's not even specific seasonal or weather-related issues. It's the dynamic range in those.
Right.
If you have big changes from year to year.
Got it. And you know what else is like that? Right over next door is Lyme disease.
Mm-hmm.
When you have mast years. Got it.
Just had one a couple of years ago. And in fact, when Vincent and I were at the Tufts Vet School, we discussed that a little bit because it has had an effect on their wildlife clinic.
There you go. So if they don't want to hear about the weather, that's their fault. But
but we're going to talk about the weather as it
pertains to viruses.
As it pertains to viruses.
Totally. And I have a really strong memory of this Four Corners hantavirus outbreak.
Yeah, I did.
Because I had just joined a virology lab in graduate school at that point, 1993.
Huh.
And this was going on and was breaking in the news. I was like, wow, I picked a cool field, didn't I?
You did.
Yes. I have a really strong recollection of this as well. And it in my recollection, it's like my first awareness of a spillover like this into humans. And I wonder whether how many previous sort of how much previous real-time documentation of a spillover like this there had been. Maybe this was sort of the beginning of the application of modern diagnostics to a situation like this that allowed us to figure these things out in real time. But maybe it's just my own awareness. I suppose Lassa fever was. Go ahead.
I was going to say not to be left out, but I also have a strong memory of this. And there was a really nice article written in December of 1993 by Denise Grady in Discover magazine, and it's available online. So I put the link to it in the show notes. And it still reads really cool, like a detective story. And then this was also shortly after Stuart Nichol joined CDC. He had been a faculty member at the University of Nevada at Reno. And I remember him later telling the story. So they had some antibody evidence that the causative agent was maybe a hantavirus or at least maybe a Bunya virus. I'm not sure how good the antibody data was, but he did PCR, and he said it still remained at that point when he was telling me about it, one of the high points in science for him, because he remembers at 4 a.m., looking at the gel and seeing the positive PCR fragment that indicated that they were really on the right track. So besides the antibody data, now they had nucleic acid data.
Right.
That was so.
A couple of other things about this are notable in my recollection. First of all, it was not a big outbreak.
No.
There were like half a dozen cases or something like that. And really, the critical thing was a couple of local physicians who, it was a devastating illness, but a couple of local physicians noted that relatives were getting this and said, something is up here, and alerted the CDC early on. Yep. So that was important.
Yep.
The other thing that I find interesting is, there had been decades of work done by field biologists who figured out the range of various species of wild mice.
Okay? Yes.
And probably took a lot of grief from people who didn't understand why anybody would want to know where the mice live.
Yeah, that's
okay?
Well, I don't know if they took a lot of grief, but they certainly didn't get a lot of money.
No.
Right. And then this breaks, and it turns out that understanding what the geographic distribution of these various mice were is actually critical in understanding this disease. And if I'm not mistaken, there are other variants of this virus that live in other species of deer mice that have appropriate distributions as well. And there's a whole history of cases of ARDS, acute respiratory distress syndrome, of people becoming very ill or dying from unknown causes that some of them have been studied in retrospect, and it turns out were the same or a similar infection, a hantavirus infection. So this has been going on forever, but wasn't discovered until this particular outbreak in the Four Corners.
Yeah.
So what are hantaviruses? They are enveloped, negative scent, single-stranded RNA viruses. The genome is in 3 segments, appropriately called the L, the M, and the S segment.
Large, medium, small.
Large, medium, small.
Small one's about 1 to 2 kb. Medium is about 3.6. L is 6.5. Code a handful of proteins.
And they're named hantaviruses after the Hantan River in Korea.
Hantan, it's right.
That's right.
Because that apparently was an okay location to name a virus after.
Yeah, sometimes it works, sometimes it doesn't.
Yeah.
Zika, right? No problem there.
Right.
So besides, as Rich mentioned, besides Sin Nombre, other strains have been isolated from different mice. One is Paramiscus maniculatus from a case in California, Paramiscus leucopus from New York case, and this is my favorite, Black Creek Canal virus, isolated from Sigmodon hispidus. Is that a rat, Sigmodon?
It is a multi-mammoth.
Multi-mammoth mouse?
Rat.
Now let's look it up. Sigmodon. No, not sigmoidoscopy. No. Sigmodon is a cotton rat. Oh, yes. Adenoviruses.
And Litisomordius carini.
Black Creek Canal.
Cotton rats are neither cotton associated nor rats, right? Aren't they?
That's exactly.
I love it. Neither cotton.
It's true. It's true.
Yes, they're not rats. Yeah. Black Creek Canal. Isn't that cool? I want to live near Black Creek Canal.
Not Dead Horse Gulch.
It's in Florida, isn't it?
Dade County. Mm-hmm. So the CDC tracks these.
Right.
And there's a wonderful map of infections by state. And so as of January 2017, this particular map shows 697 cases in 32 states. The biggest one, Colorado, looks like. No, it's New Mexico, 109. Colorado, 104.
This is cumulative cases.
Cumulative.
Right.
Okay, so I guess that's cumulative. They don't say over how long a period of time, but probably since its discovery, you suppose.
Yeah.
I mean, it's not
93.
It's not something that's,
93 in the U.S., yeah.
So the other thing that's important here to remember is that not only is everything connected, the wildlife is connected to the plants.
Sure.
So the plants, their distribution will determine largely where there's a lot of pinyon nuts and not.
Yeah.
And that has a very, very big outcome with response to disease spread.
And because this is a global virus world, there are also international cases of HPS in many countries. I'm looking here, mostly South America, right? Argentina, etc. Canada. Let's see if we get outside. Yeah, that's all. Okay, international cases. And then I wanted to mention, we've actually mentioned these on TWiV. There have been cases in California and New York associated with camping, right, in these wooden structures or cabins.
Yeah, these are I think they were like luxury tents, so I guess it's glamping.
Platforms. They're called
I don't think they were ever called luxury, even in Yosemite.
Okay. Okay. I thought the deal was that they were these double wall insulated tents that were somehow somehow an upgrade over your usual.
But
I don't
the point
know.
is the mice were living in the
mice were living
National
in the walls.
National Park Service here. I don't.
Well, right. Okay. Yeah. As glamorous as the National Park Service gets.
There was a big. They created Adirondack State Park or National Park, and in doing so, they forced all of these people that had cabins on wooden platforms. They were called platform tents.
That's right, platforms.
To get rid of them, and you can't believe the slanderous signs regarding the National Park Service by the locals that were absolutely livid about not being grandfather claused into this thing, and they all had to get rid of them. And there were many, 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 many of them.
In New York State, there have been 5 cases since 1993, 3 in Long Island, 2 upstate. And this past summer, someone died at Belmont Racetrack, and they think it was a hantavirus infection.
Wasn't there one in Queens also?
That was West Nile.
Speaking of Queens,
yes,
there was, when I was looking through Discover, I also found an article that I had saved about the sweating sickness that maybe Anne Boleyn had survived. And these 2 investigators in 1997, Vanya Gant and Guy Thwaites, in London, thought that maybe she and others who had one of 4 different times of a recurring disease called sudor anglicus, or English sweating sickness, that the symptoms really matched one of these hantaviruses or hantavirus pulmonary syndrome, where you've got a headache in the morning, you're short of breath in the afternoon, you take to your bed at tea time and you're on a respirator by midnight. Well, not in Anne Boleyn's day, but anyway. So
she found a definitive cure for the headache.
She did, yeah.
Right.
That's right.
Definitely. But
the one body problem moved to a 2-body problem.
Right. And so they even identified somebody who succumbed to the epidemic in 1551, and they know where he's buried, but the possibility of getting hantavirus RNA out of an exhumed body probably was beyond the finances available. So anyway, I thought it was kind of a neat thing. It kind of goes back to us saying that, this disease or many diseases have been around a long time. It just takes a cluster of them, or as Rich said, some people to notice and put 2 and 2 together and find this, that maybe this goes back even to England.
So we have had letters to TWiV about sweating sickness. A couple times people have asked, have you ever talked about sweating sickness? And people have sent us links and so forth. Yeah.
Actually, an important feature of this disease that we haven't discussed is that it's not transmissible from human to human.
Mm-hmm. That's right.
And, that really affects the dynamics significantly.
Mm-hmm.
Yeah.
Although I think someone should try and develop a transmissible strain, don't you?
Yeah, right. Absolutely.
Maybe in ferrets or something.
I was just going to say they should use ferrets. Yeah.
Ferrets.
Now there are other hantaviruses that cause different diseases. One is hemorrhagic fever with renal syndrome, HFRS, and as that's opposed to FRS, Fellow of the Royal Society, right? The 4 viruses that are associated with HFRS are the striped field mouse, Apodemus agrarius, that's Hantan virus, Norway rat, and the black rat for Sol virus,
Rattus norvegicus and Rattus rattus.
Rattus rattus. This is Dickson's favorite one, right?
Rattus rattus.
I like both of them.
I like the the species names that are like that, like gorilla.
That's right.
I don't know if you know this, Kathy, but Dickson, a long time ago, told this story of how when he was young and living in New Orleans, he was bitten by a rat.
Oh, yes.
Oh, I have heard that story.
Oh, yeah.
Was that on TWiV, maybe?
I got the scar to show it.
And we always...
but it wasn't radioactive, so he didn't get any superpowers.
This is all true.
Whenever I see rat, I think of you being bitten, Dickson.
Well, thank you.
And the bank vole, which is a very wealthy species.
Yes.
For
they're found in Switzerland.
Pumala virus. And this is good. The yellow-necked field mouse for Dobrava virus. So these cause HFRS, renal failure that happens before pulmonary edema and disseminated intravascular coagulation.
DIC. Mm-hmm.
Mortality 5 to 15%. A more moderate form caused by Sol virus.
Disseminated intravascular coagulation is just your blood clots.
Mm-hmm. Yeah, this sounds really unpleasant.
Really, really bad.
It is.
Thousands of Eurasian cases of Sol virus annually. Serologic evidence for infection with Sol-like hantaviruses has been found in major cities of the U.S., in rodents, that is.
That's right.
Recently implicated in human cases of HFRS in Baltimore. And I think Pumala virus, we talked about not long ago. Someone asked us about it. A mild form of HFRS is caused by Pumala. It is responsible for nephropathia epidemica in Scandinavia, and mortality rate of 1 to 3%. Nephropathia epidemica. Interesting name.
Yeah.
So there are that's your outbreak for today, for this month, actually. And there are a few more, but I noticed that the outbreak exhibit hantaviruses did not get a lot of space. There was more space for Zika and Ebola. Ebola had a huge space, but Hanta, very little, but they're pretty interesting.
And it seems like we're more likely almost to come into contact with hantaviruses than Ebola, at least in North America.
Yeah.
So I always say, if you come into contact with, if you see any mouse droppings, mouse pellets, don't inhale them.
Exactly.
No. Right.
You shouldn't do that.
Well, and in a more practical sense, if you're cleaning out the shed, or I keep bringing that up because we have a mouse problem in the outdoor areas here. But if you're doing some kind of cleaning work and you see the mouse droppings around, don't just sweep them up with a push broom. Pay a little attention. Try not to raise dust. Get some ventilation through.
Absolutely.
Maybe put on a mask.
Don't want to aerosolize them. Right.
Right.
All right. Now we have a completely different subject. We're
still in mice, though.
Mice, and we're going to switch to the
Still
GI.
Viruses.
We're going to always viruses here on TWiV, but we're going to switch to the GI tract.
No weather in this one, though.
But yesterday on TWiV,
I wouldn't say that.
I picked a paper in honor of Dickson. I have to mention this.
Yes.
Which involved parasites that infect trout.
Right.
Those are two of your passions, right?
They are.
Oh, the only two of your passions.
Well, no, I have many.
Yeah, okay. You say that. Keep saying that. But anyway, trout and parasites are pretty
He's
cool.
clearly into photography.
Yeah.
Are you?
Yeah, absolutely. Okay.
If he's not into it, he's really good at something he's not into.
Well, that's very kind of you.
Intestinal dysmotility syndromes following systemic infection by flaviviruses.
Oh, wait. Cell is a U.S. journal, so they're intestinal.
Yeah, I know. I like saying the other pronunciations. Kathy, I was talking to someone recently, and they said, what's the word that you and I have agreed on the pronunciation? Niche. Niche.
Oh,
niche. So they said niche, and they stopped a few minutes later, and they said, oh, it was Nels on TWiV. He said, or is it niche? I said, well, it's niche. Yeah, Nels.
Just remember, if you have a niche, scratch it.
So this is in Cell. And it comes from Washington University School of Medicine, Yale School of Medicine, and University of Pennsylvania School of Medicine. We have, is there an equality in the first authors? I don't see it here, right? Just a sole first author, James P. White, Zhang, Malvin, Curry, Honold, Hookeroth, Stappenbeck, and Michael Diamond. And this is all about intestinal or intestinal disorders.
Good.
Dysfunction in dysmotility of your gut. 10 to 30% of Western peoples suffer from some kind of intestinal dysmotility.
Not moving the way it should.
Not moving. And a major subset is irritable bowel syndrome, IBS, 10% of the population. And there are others that are more rare. And they often seem to follow inflammatory events. Now, as a little backdrop for this paper, in our gut, down there, we have a nervous system.
Oh, yes.
The enteric nervous system.
You know where that was discovered?
Right here.
Got it.
By Gershon, Michael Gershon. Did he discover it or just work on it?
No. He characterized it along with several of his graduate students, MD, PhD students.
Uh-huh. So there's a big network of neurons and glial networks down there, like the myenteric plexus.
And it's not connected to your brain either.
It's not?
No.
How can it not be?
Well, there's...
works through the vagus system.
Well, that's right. Not directly.
The vagus nerve connects it, and there's increasing evidence that connection is a lot stronger and more direct than was previously believed. But you're correct. It was not believed to be connected. It was thought to be independent, but it is in fact a lot more connected than we thought.
So myenteric plexus,
that's right.
Submucosal plexus, and these control gut motility, right? The speed at which things, and also the function of the epithelial, blood flow, responses to sensory stimuli, etc.
And your feeling of well-being.
Oh, I must have a good ENS then.
You have a gut level feeling for this?
I always feel good.
Yes.
So the idea is that dysfunction or degeneration of this system underlies these intestinal dysmotility disorders. Mm-hmm. So you may be thinking, what does this have to do with viruses? Well, as we know, there are neurotropic viruses.
Well, golly, Vincent, why should you say that?
And, one of the viruses they use in this paper is West Nile virus, which is a mosquito-transmitted flavivirus that made Dickson famous, actually. And a fraction of human infections involve brain infections, right?
Absolutely.
Meningitis, encephalitis, sometimes death. So the virus is neurotropic. And there are other flaviviruses that are neurotropic, like Zika virus and Powassan virus, a tick-transmitted flavivirus. But can they infect enteric neurons, right? That's the question. Well, apparently it's known previously that if you infect mice with some of these neurotropic flaviviruses, you get GI tract pathology. And even in birds, West Nile infected birds, they see some GI pathology. Remember that, Dickson?
I do.
You wrote the book.
Well, you helped.
West Nile story. No, I didn't do it.
You corrected all the virology.
No, and people who
this is consistent with the human data too, because there are a lot of case reports of people with GI symptoms with these viruses.
Right.
That's right.
Now, so there's also a history of research on infections of various sorts triggering these intestinal dysmotility disorders, not just viral, but bacterial and protozoan as well.
Trypanosoma cruzi targets the myenteric plexus, and that's why you get these large organ diseases from that.
Megacolon.
Megaesophagus. That's right.
Right?
Exactly right.
So that's the background for this. And they said, well, let's infect mice with these neurotropic flaviviruses and see if we have gut issues. Right. It's straightforward, and it's really very interesting, I think.
Sure.
So they infect mice, the wild-type mice, C57 black mice, with West Nile virus or Zika virus, and later on with, Powassan? Yes, the tick-borne virus. Now, go ahead, Kathy.
I was just going to say the way they do it is through the foot pad. So they're doing a subcutaneous injection.
Right. And for Powassan, they do it a different way, right?
For the Kun one.
Kunin is IP?
Yeah.
Okay.
But Powassan, I think, is also sub-Q.
So I blogged about this last night.
The Kunjin virus is an attenuted variant of
West Nile.
WFP. Yeah.
So I had blogged yesterday, and I wrote it was orally given because I figured, they're looking in the gut.
How else do you get it there, right?
But So the idea is that it's injected, which mimics the natural route of infection by a mosquito, right? And then you have a systemic infection, which of course can deliver virus to the intestine because vascular system. So my bad, and Kathy corrected it at 4 a.m. this morning. Or
No. That's when it got uploaded.
I know. I'm just kidding. But thank you for the correction, because I assumed it was oral. And you're right. They don't actually say it in the main text.
Or in the figure legend. It had to go
should.
to the materials and methods.
Yeah, you really should say. It's an important
That is an
thing,
important
right?
point.
Anyway,
And it's interesting, too, because these are viruses that are normally transmitted not orally.
that's right.
So this is kind of it's important to point out.
I was lying awake at 4 a.m. and saying, how did this virus get through the gut? It's pretty interesting. It's envelope, but it's not through the gut.
Well, T. cruzi doesn't go down the gut either. It goes through the mucus membranes and gets into the blood supply, right?
Yeah. So anyway, they infect mice with West Nile, and by 8 or 10 days after infection, you see dilation of intestinal segments. I mean, they pull the intestine out of these mice and they put them on a piece of paper. You could see.
Yeah.
You do not have to be a pathologist to see this.
Here.
Parts are bigger. Not the whole thing, but the parts have expanded.
It's actually really pretty dramatic. I looked at this. I was like, oh wow, yeah, that is definitely dilated.
We note some degree of regional specificity of dilation, like regional airlines, right?
Right.
So it's the same thing with Zika virus infection. So the question is this a consequence of virus infection, right? And I love this part, and the all of you on TWiV will love this. We measured viral RNA in the gut because infectious viruses are inactivated by bile acids. So they're thinking of us.
I felt like they were. Yes, I felt that was put in there for TWiV.
Probably not, because Michael Diamond's a good virologist, but there's a reason why you can't do plaque assays here.
Right.
So they can detect as early as day 2 after West Nile viral RNA in the regions of the GI tract of some of the animals. And by 4 days, all mice are infected in all regions. RNA levels peak at 6 to 8 days. And the highest, then they make a slip here. The highest viral titers are observed before declining 21 days. Of course, they're not viral titers, right? But they could see RNA through 56 days in all segments of the GI tract. So it's a long time, right? It's more than a month.
And that, in a mouse lifespan, is quite a while.
Exactly.
And Alan, what is the lifespan of a mouse?
In captivity, a couple of years?
Yeah.
2.
Mm-hmm.
What is it in the wild? It could be a day.
Depends. Depends on whether you're an owl or a hawk or
Exactly.
Yeah, they get them.
They do.
So West Nile and Zika do this. And so then they looked at motility, bowel motility. I guess they feed the mice a fluorescent dextran, fluorescently labeled dextran.
Yeah.
And they see how long it takes to go through into the colon, I guess.
It got transit time.
Got transit time. And it was slowed in many of the infected mice.
Interesting.
It looks to me from I didn't read the methods on this, but it looks to me from this as if they feed them and wait a certain amount of time and then sacrifice them and find out how far it went.
Yeah.
These graphs are fascinating. The x-axis is essentially a map of the GI tract from the stomach through the small intestines and cecum to the colon. And the little bar graphs show how far under each condition that dextran made it in a certain period of time.
Right.
Mm-hmm. So that was West Nile and Powassan, and I think also quinine. Don't remember that they see similar effects.
Mm-hmm.
So the question is, would any systemic viral infection do this? So they pick chikungunya virus, which is not usually neurotropic. It's arthritogenic, causes arthritis.
Different family of viruses. It's an alpha virus.
Yeah. How are these?
It is mosquito transmitted.
Mm-hmm.
Yes.
Sub-Q.
So that
So they measured RNA levels, GI transit, and much lower virus in the GI tract, and no intestinal dilation, no alteration of GI transit. Right. So, at least for chick, You don't see this.
How about poliovirus?
They say systemic viral infections, therefore, do not generally dysregulate. Well, I would say chick doesn't, right?
Right.
I don't think you'd say it for all viruses.
Polio?
Yeah, Dickson,
Polio?
actually, reading through this whole paper, I kept thinking, there's a neurotropic virus that goes through the intestine that I'd really like to see this done with.
Yeah, for sure. Any enterovirus, in fact.
Yeah.
Except the rhinoviruses, which are part of that group now, but the ones that actually infect the enteric tract, as we'll see, they're probably more likely to be associated with these syndromes in humans because West Nile infection is pretty rare.
Not very good. It's not 10% of the population.
Yeah.
But a lot of nematode infections in gut tract slow it down. A lot of them.
Oh, yeah?
Yep. Trichinella is one of those.
Do they infect the enteric nervous system?
They don't, but they secrete things that interact with the nervous system. Like acetylcholine.
So my understanding here is that the transit time is affected by the absorption. So if you don't get absorbed, then you remain in the intestine. But if you're absorbed, then...
But dextran can't be absorbed.
Right.
Because it says here, dextran is poorly absorbed, so luminal concentrations provide a measure of GI transit. That's what I was wondering.
You know what? They used to give charcoal.
Yeah, right.
They used to just inject a solution of suspension of charcoal or India ink. And then you could just open them up and see exactly where the pellets are.
The fluorescent dextran is kind of convenient.
Yeah, it's nice stuff.
All right, so where is the virus replicating in the gut? So they infect and they do staining for viral antigens and a neuronal marker.
Good idea.
So, and they can find West Nile proteins in ganglia of the myenteric plexus of the small intestine and isolated neurons, but not in the epithelium of the GI tract.
So, interesting question here. This is not just a virus of vertebrates. It's from the mosquito too, right?
Sure.
So I wonder if it infects the ganglia of mosquitoes as well.
You don't know the answer to that?
I don't know the answer to that.
Someone must have looked.
Well, these guys looked for the first time here, so I think maybe...
You want to know if any...
Yeah. Is it neurotropic no matter what?
Oh, well, we could Google that.
We could.
Is West Nile virus neurotropic in mosquitoes?
Right.
Let's see if we get quack sites.
Those are ducks.
Just now, it doesn't say.
Probably
I'd have to look more carefully.
Yeah, because this is one of the most promiscuous viruses.
I'm sure
that
someone
I know
is
of.
yelling right now. Yes, no.
I'm looking on PubMed, but it's going to take me.
So they say that West Nile preferentially infects enteric neurons. It's
preferentially
in the gut.
There are receptors there, right?
Well, there must be, but it's very interesting that no one looked before.
It is very interesting.
Although they did have, as we said before, some evidence in people.
Yeah.
What else is interesting here? So I think they also see infiltration of immune cells into the infected area, but they don't see any alteration of the villi, no blunting, no hemorrhaging, no cell death, which you would see for other intestinal viruses like rotaviruses or noroviruses.
Right.
But the neurons did display nuclear hypertrophy.
Uh-huh.
Mm-hmm.
So something's going on. A little bit bigger.
Yeah.
And then they ask, what is going on in terms of immune cells in this? Is there some infiltration? So they stain for T cells, and they could see that there are T cells coming in. Initially, the numbers go up, and then by 10 or 28 days, they go back down again in these same areas of the intestine. And they seem to be West Nile virus specific CD8 positive T cells because they can stain for those. They could stain for a specific peptide that's known to be a West Nile CD8 T cell epitope. And you can see that they're going in. So infection is stimulating infiltration of virus specific CD8 positive T cells. And this whole thing is associated with cell death, apoptotic cell death, which they look for specifically.
Right. So CD8 positive cells are going to kill.
What, Dickson?
These are cytotoxic.
I'm thinking about the epidemiology of this infection and the role that birds play in the transmission cycle. And they did a lot of studies in crows. Cornell has a big center for West Nile.
Can you make a crow sound?
I could, but I'd rather not right now. I don't want to offend too many people.
Someone did.
It might stick in someone's call.
Exactly.
Someone did, and I put it at the end of the show. It was you, I think.
I think I
You did a
that.
good call.
I can be cajoled into doing this later,
Sorry.
but so the point is that on day 1 of infection with crows, they're perfectly normal and they can rook, and they can forage along the side of the road. The second day they're lethargic,
Yeah.
but they're still flying. The third day they can't move.
Okay.
So what does that do? That immobilizes the crow and it makes it an easy target for mosquitoes.
Yeah, you think so?
Yeah, I know so.
What, you think a crow could outfly a mosquito normally?
Yeah,
mosquitoes
sure.
are pretty fast.
Yeah.
Yeah.
But no, mosquitoes don't fly all that well.
They
Okay.
They
Okay.
don't.
Yeah, that's interesting, isn't it?
So this is probably some biologically significant.
May have been selected for evolutionarily, right?
Exactly right.
Yeah. Right.
Now, in raptors, my understanding is that West Nile tends to affect their vision.
Which would be
So
That's interesting.
neurotropic.
Yeah, it's a different a different pathology in raptors than it is in crows. Crows will end up just kind of sitting around, and raptors will just be unable to hunt.
So, Alan, what is the main effect of a raptor being infected by West Nile? They forget the words to their songs. They
Back
lose
to
the beat.
the science. So we've shown that there's an infiltration of virus-specific T cells. There's cell death of neurons in this area. So they want to know, are the CD8 T cells responsible for this? Okay. So they take mice that don't have these T cells. They actually don't have B. A variety of mice, which 3 different strains. They have no B cells, no T cells, no alpha, beta, and gamma delta T cells, or only CD8 positive T cells.
Amazing.
And they basically can conclude that this T cell infiltrate contributes to gut dysmotility.
The CD8 positive T cells.
CD8 positive T cells.
Because the CD8 knockout mice were also affected. Did not get the gut dysmotility, and neither did any of the others, which also lacked CD8. So the CD8 T cells are the key to this gut dysmotility thing.
CD8 T cells. Yeah.
What was the mortality rate for this? They must have mentioned something about that. I'm sorry that I didn't pick up on that.
What is it? I mean, not every mouse dies in their experiment, but I think I don't know the number. Do you know? Anyone know? I don't recall.
No, they showed survival curve of something in the supplemental data.
Supplemental data. There you go.
It looked like about 50% died in this experiment where they also gave the anti-West Nile virus human antibody. Those survived, but the ones that were the controls did not. And it was about 50%.
Okay.
Right.
So let's see if it tells us here. They did some longer studies.
Yeah, there were some studies that they couldn't do over a length of time because the virus infection was too lethal.
Now, the Kunin is attenuated, so they live, more of them live after infection with that virus. Mm-hmm. Most West Nile infected mice showed delayed transitive die during the acute stages, but infection was maintained as far out as 4 and 7 weeks post-infection. Those surviving animals had persistent dysmotility.
Interesting.
Even though virus is starting to go down.
Another thing they did in some studies at Cornell to show that the mosquito is not a necessary vector for West Nile was that they took an infected crow and they put it with some uninfected crows.
They could transmit it to
Eventually, they all got sick.
They are aerosolizing it,
So
or biting
it was
each
in
other?
the secretions, the nasal secretions.
Interesting.
And they share that. They're very social animals. So as a result, that was another interesting nuance here that you wouldn't have guessed at.
So here's the thing, Dickson. Mice that died developed the most severe delays in GI transit during the acute infection. And they could prevent that by treating the mice with an antibody, a monoclonal, against West Nile, which they would give them 4 days after infection, and it could prevent death. And they didn't develop the GI dysmotility. So, and this last experiment is very cool. So in people, apparently, these dysmotility orders are intermittent, right? They're episodic.
Right.
And you have it. I know of some people that have this, and their whole life they have episodic. And it seems to sometimes it corroborates with an infection or some kind of inflammation or even stress.
Right.
You trigger these. So
Yeah,
they said, can we duplicate this in mice? So they took the mice that survived West Nile infection, and then they gave them either a Venezuelan equine encephalitis vaccine strain, right, which is not going to hurt them, but it is going to infect. It's an attenuated vaccine strain, or synthetic RNA poly-IC, 8 weeks after West Nile infection. And they could see a reduced gut transit, bowel transit after treatments, 4 and 8 weeks post-infection, slower transit, which is not, caused by immune infiltrates, additional cells coming in. So it's a different kind of trigger. So in other words, the mice were somehow sensitized after West Nile infection. And then when you give them another virus or synthetic RNA, it's doing something that is triggering another bout of intestinal dysmotility.
Right. And mechanistically, they don't understand that.
They do not, because it doesn't involve T cells.
It's not the T cells that time.
Yeah. It's really interesting. So, in people, you could have an early infection of some kind that sensitizes you. Maybe it's even a chronic infection that doesn't cause any issue in other people, but in you're now sensitized to episodic bouts of IBS.
It seems like they also can't quite rule out some kind of West Nile virus persistence.
That's right.
Right.
That's right.
They say that it might be involved in chronic GI dysfunction.
Yeah, so I mean, it could, and then in humans, that's why I'm thinking if it's an enterovirus, it could persist because people are always infected with enterovirus, and we don't have enough data to say if it occurs for years and years and years or not, because we're always full of enteroviruses. So
remember that for every sick person that this induces illness in, 200 other people are asymptomatic. 200. So imagine the size of the epidemic. Yeah, for West Nile. So they've had epidemics where 3,000 or 4,000 people were sick. You multiply that times 200 and you've got 600,000 people now. And now I would love to see the epidemiology of irritable bowel syndrome as a
and look at viral serology, right? Yeah, they talk about that. They need some really big studies. Now they say that it's unlikely that these flavies are involved, but your point is very well taken. A few 100,000, it could contribute to it.
Absolutely.
Add to it. And it seems what they're getting at here, and what I think is probably going to end up being the case, is that this is not WNV specific. That's one of many that can trigger this sort of thing. And what we're looking at is an immune response gone awry. You get CD8 positive T cells in the gut nervous system, and they start killing or damaging cells, and then that causes this persistent problem, which then flares up again by some other mechanism that remains to be figured out, and that's a mechanism, or maybe the mechanism, for things like IBS.
So I had a friend who worked on trichinella and worked on the motility aspect, right? His name was Gilbert Castro. He was at the University of Texas at Austin. And he used to do electrical measurements of the conductivity of various segments of the gut tract as a measure of efflux of ions that were induced by the trichinella infection. And all of the wheat germ agglutinin receptor sites were gone after trichinella infection. They never came back. That's a very, very common receptor on the gut tract epithelium. And just one infection wiped out the whole thing. Now, what effect would that have on the future function of that gut tract? I mean, do you absorb the same way for your amino acids and fats and
surprising. I thought that the gut cell, the epithelial turnover would restore it.
Exactly
right.
Right.
It does not?
Nope. It's gone forever. So it's probably being affected at the base of the crypt, which is the source for all the new cells that are coming up. And there's a cell there called the cell of Paneth, which has a very unusual functional aspect to it and very different. And I think we don't know a lot about the gut tract that we should probably pay more attention to later because people are making a living out of treating these illnesses in ways that probably have nothing to do with what's wrong. That's my suspicion, at least.
So let me tell you what they say here. We wish to highlight that neurotropic flaviviruses are unlikely to be the major cause of common GI dysmotility syndromes in humans like IBS because of their low incidence. our data suggests rather that systemic infection by other viruses, and they say enteroviruses, which I think
Woohoo!
is great because they cause millions and millions of infections,
Sure.
many of them asymptomatic.
Yep.
And they also say herpes viruses, astroviruses, and other flaviviruses. So I think enteroviruses are a great candidate. But then, you can imagine you get infected at a young age, and it just predisposes you to these recurrent bouts of IBS caused by stress, inflammation, whatever, right?
After they treated the mice with the antibody to cure them of the infection and save their lives, what was the gut function like after that?
Apparently, I think the motility was good.
Yeah, but maybe the absorption qualities weren't, or maybe there's some other effects.
That was the one they treated shortly after infection, so the infection didn't really get going, and I think they just didn't get this response at all.
So I'm concerned that the myenteric plexus is a very essential portion of the gut functionality, right? Mm-hmm. And nervous tissue repairs itself very differently in some certain circumstances. How does the repair mechanism look for this one? And does it?
Mm-hmm.
Yeah.
So what I'm thinking also right now is people who had polio. I wonder if they are predisposed to IBS too.
Right.
We always say the gut phase of polio is asymptomatic, doesn't do anything, but maybe it's involved in this. A lot of people had polio, and remember, not just the paralytic, but 100 times more non-paralytic polio. You could do an epi study on them, right, to see if they're predisposed.
That's right.
But remember, enterovirus infections, there are millions and millions just in the U.S. every year.
Yeah.
Many gut infections, but other parts of the body as well.
We've all had enteroviruses.
And I could easily see them being involved in this.
So then the model would be that this is, most people get an enterovirus, nothing happens or very little happens. But then something goes wrong in 10% of the population.
Exactly.
And you get this type of involvement.
So that's the other question. Go ahead, Rich.
I wonder if the something that goes wrong has some sort of genetic basis.
For sure.
And I wonder whether, somebody must have done genome-wide association studies on populations of people with irritable bowel syndrome.
Yes.
Right.
And I wonder if that would turn up some appropriate association that would be relevant to a virus infection.
Yeah.
Right.
I would bet you get changes in immune genes. But, there'd be a lot. And you'd figuring out.
Yeah, this is going to be so multifactorial. The trouble with GWAS studies is that you're often looking at a bazillion different things that can contribute, and then you have a hard time with working out the statistics. The other thing that occurs to me, though, is this could be partly genetic. It could also be partly microbiome related.
That's Alan, you took the words right out of my
Oh, well then.
frontal lobes.
Yeah. So we're talking about a system that is just it's the ancestral homeland of the microbiome. This is where they all are.
That's right.
And that's, I think, going to play a big role in this.
So we've got an epidemic right now, which is not localized.
Of microbiome papers?
Of obesity. Obesity. Yes. And it's linear almost. From the time they started to track this back in the late 80s, and they isolated it down the Appalachian chain of command, so to speak, all the southern states that had high fat intakes and bad diets and all this, they were blaming it all on that. But now this epidemic has spread to almost every country, and it's up to 40% of the human population are overweight.
There's some countries that have avoided it.
Some, but not many. So the point is that a linearity of this kind of thing, this is a cumulative thing. It doesn't go away. Once you get something, it stays with you. And I'm concerned that we're really overlooking something important with regards to this.
Let's do an experiment. Let's shut down all the McDonald's. See what happens.
I don't think that's going to work at all.
You don't think so?
No, I don't think it's from that kind of
still
at
Burger King.
The point is that people are gaining weight at a rate that has nothing to do with their diet because they're eating the same things.
This is a really complex topic.
I've sat through
multi-day meetings on this.
This is beyond this podcast.
People are not eating all the same things and getting equally fat. It's there are differences there. There are differences in exercise. Everybody points at different cause, and that's going to.
They're all wrong.
There are some people who point to a single cause, but I don't think that's going to be the answer.
Kathy, enteric adenoviruses. Could they be involved too?
I would think so.
Now, when you get do you have an overt gastroenteritis or are there asymptomatic infections?
Good question. I think they must be symptomatic, or else how would we have even discovered them?
I mean, there are
I
some.
could be wrong. Yeah.
There are some symptomatic adenovirus infections. The question is, are there a bunch that are asymptomatic?
Yeah. I think a number of gut viruses could be participating in this. And that makes it harder to tease out, right? Because if you do the epi, and they make this statement, which is really interesting. I think it's the last statement of the paper. Oh, I always like to read it here. Mm-hmm. Ultimately, establishing the roles of neurotropic viral infections in the development of human GI transit disorders will require a rigorous, multi-institutional, and likely large serological study with well-characterized affected cohorts and matched controls. We need a lot of money for this.
Yes.
It's going to be a complicated study because I would say many different viruses are going to be involved, right? And if you're going to look at the genome, the microbiome as well, pretty complicated. You're going to need a big computer.
Yeah, and you're going to get output.
We're going to need a bigger grant.
You're going to get output that's the same as the GWAS studies, with it being multifactorial. It's going to be it's going to be hard to get it funded, given the probable outcome of something.
Well, but I think this paper sets out a marker with a really, really powerful animal model.
Right.
Yeah.
Because you've got a system where you can look deep into mechanisms, and it seems to be really strongly parallel to a lot of what you see in humans. So
yeah, I like it. I liked it a lot. And in fact, Alan, you had originally said pick a simple paper,
right?
So this was my simple paper.
This would have been great. Yes.
Okay, so quickly scanning, I found that non-cultivable adenoviruses in the stools belong to group F, serotypes 40 and 41, and these are associated with diarrheal disease in children.
Mm-hmm. Okay, so it could that could be the initial infection, right? And then something else causes your IBS over the years.
So that's right. You're looking for a before and after, and you never know what the before looks like because you're always dealing with the after. And that's tough to know where that occurred.
Well, here's the problem. You're going to do a big sero study, and you're going to see, all these people have enterovirus, and 10% of them have IBS. What is it? No,
you won't. No.
Because everybody has enterovirus, and 10% of the population has IBS.
Right.
Back where we started.
Yep.
Anyway, I really enjoyed that. I thought it was cool.
Good stuff.
Good stuff. Good example of when an animal model is valuable. It's not telling you exactly what's happening in people, right? It may not be that virus, but it gives you some clues.
With West Nile, there is no animal model. They're all valid studies for that particular species, right?
Here's some crows.
Right, and this particular animal model and this work points the way towards dozens of additional experiments that you could do that would be informative. So it's going to keep somebody occupied for quite a while.
Sure.
All right, we have a couple of emails that we shall do. First one is from Steve, who writes, thought you might be interested in this snippet that turned up in my weekly updates from our UK Health and Safety Expectorate.
Inspector.
No.
I wanted to see if you're listening.
Yes.
I don't remember, and Rich is listening. Good. There being a publicized national scare about it, and it doesn't say what the hazard level was, but it's sobering to read of emergency backup generators failing and even catching fire. All the best, Steve from Luton, UK, where it's cold, windy, and pouring with rain.
Right.
So apparently in September 2014, the mains power was lost at a site including high containment laboratory facilities. Probably a BSL-4, I guess, or maybe BSL-3. And power was lost. 12 generators failed. 2 failed to operate. 2 started but failed. 1 of them caught fire. Emergency escalation system was triggered. And they lost power for a few hours. So he sends an article about this. But it sounds to me that it worked in the end. They controlled it, right?
That was my reaction to this. Even though there were some failures in the emergency power system, it came out okay. They had enough backup so that everything came out okay. Now they say that it happened. It was lucky that it happened sort of off hours, and there weren't any serious pathogens being worked on at the time. And had circumstances been different, there could have been a problem.
And apparently, as a result of all this chaos, all power was lost to several of the high containment facilities for several hours, which, that shouldn't happen.
Right.
Well,
so
sure
nobody died apparently, but that doesn't mean that everything was okay.
The company that was involved in providing the backup generators has been fined. That it just popped up in December 2018 news, which is probably how Steve ran into it.
So this news item starts off: Hove Crown Court heard how yada yada in 2014. So this is apparently a court case that is going on. And that's how this came out. And this was not made news at the time it occurred. I don't know what the regulations are in the UK, but if you have an accident like this at a containment facility in the US and it doesn't get reported, that in itself is a problem.
Mm-hmm. Yeah, it has to be reported. And from what we saw at the BSL-4 in Boston, they're pretty strict about that. And they showed us all sorts of backups. And, they have generators on site and they're off site and all that. So, at some point it may happen that, you're going to lose power, but presumably you have other systems in place to reduce anything happening.
Redundancy of function.
Yeah. Alan, can you take the next one, please?
Next one is from Samir, who writes, "Thank you for the discussion of MKPV on TWiV 520. It looks like the combination of metagenomic analysis and RNA in situ hybridization will help identify previously unrecognized viral infections in animals commonly used as model systems of disease, or to understand basic biology." And then provides a link to a preprint article that used a similar strategy to identify a novel picornavirus in zebrafish, an animal model growing in popularity.
Let me interject that this is from Eric Delwart, among others, who we've covered before. And this reminds me of a former student of mine, Brian McDermott. I don't know if you knew him.
Oh, yeah. I knew Brian. Yeah.
So he did a polio project in the lab, and now he's a PI at Case Western working on zebrafish. So I have to send him this, see if he's excited about a convergence of his interests.
Oh, cool.
Go ahead. I'm sorry.
Okay. I'm glad to hear Brian's doing well, too. I'd like to follow up on comments made during This Old Mouse regarding the rederivation of mouse lines. Rederivation can be performed through a variety of procedures, most commonly either via embryo transplant into the uterus of quote-unquote clean pseudo-pregnant recipient females, the way it was done in the MKPV paper, or via cesarean section of a late-term pregnant female with cross-fostering to a clean lactating female. In the latter case, this assumes the agent you are trying to eliminate is acquired at the time of or after birth and not vertically transmitted in utero. Embryos can be harvested at 0.5 days after confirmation of copulation plug in a mouse breeding cage or developed via IVF. Embryos or sperm can be cryopreserved and banked for later use instead of maintaining a live animal breeding colony. As you mentioned during the discussion, Rodinger et al. Described elimination of MKPV using embryo rederivation. I may be wrong, but MKPV may have been inadvertently eliminated from the vendor colonies because their breeding stock is routinely rederived into the clean breeding facility. I believe murine norovirus, MNV, was not found in many vendor colonies after it was discovered because it was already eliminated during the rederivation process to eliminate other known viruses. However, the murine norovirus remains common in academic settings because of less stringent biosecurity measures and the determination by some investigators that it doesn't interfere with their research, or it's too expensive and time-consuming to do it, to rederive meaning. I wouldn't be shocked if the same was true for MKPV. I agree with Vincent. It would be interesting to know the prevalence in wild mice. Parvoviruses are notoriously resistant to disinfection, so carrying into the barrier facility would be easy if the facility biosecurity isn't tight. And Samir is a veterinarian.
Whom I met at Tufts. Did you meet Samir?
I think maybe.
He came up after the TWiV and introduced himself, and I remembered he had sent this email.
Oh, yes, right. Okay. Yeah, I met so many people that day, and I'm terrible with names, but yes, I do remember this.
Sounds to me like maintaining a commercial mouse enterprise is difficult.
Oh, yeah.
Oh, yeah.
That's something I'd want to do. We have a lot of trouble with mice here.
Lots.
lots of contamination. Aren't they rebuilding our mouse facility now?
I hope so.
On the top floor, they're totally gutting it and redoing it because wild mice were invading it.
Right.
And introducing wild mouse viruses.
There was a lot of Airbnb going on up there, I think.
Airbnb?
Yeah.
I was trying to find out more about what level biosecurity that Hove Trial Center had. And Hove Trial Center also goes by the name as Hove Crown Court. So the headline about court doesn't necessarily have to do with the legal part. It's part of their address.
Okay, sorry.
Court directors.
Right.
All right, time for some picks of the week. Alan, what do you have for us?
Okay, this is a video. It's a short video from the European Southern Observatory. And like a lot of things with astronomy, it's one of these you should put it on full screen, highest resolution you can get.
Yeah.
Let's see. Oh yeah, you can do this at 1080 high def, and just stare at it and take in the enormity of it. They start with an overview of the view of the Milky Way through this observatory's telescope, and then they zoom in and zoom in and zoom in, and they're zooming all the way to Sagittarius A, which is believed now to be a black hole, a massive black hole at the center of our galaxy.
Wow.
And then they at a certain point in the video, there's a cut that they go to an animation built from several exposures, obviously, and this model of the stuff orbiting this black hole, and it's just really cool.
How far away is this from us?
A long way. Parsecs and parsecs.
Is it the nearest black hole?
I don't think so. This is the one at the center of the galaxy, so I'm thinking
it's a supermassive black hole.
Yeah, it's
all right. So if I went near this, what would happen to me?
You'd be spaghettied.
Really?
Right.
Shredded?
Yeah. No, you'd be stretched out.
Something like your feet would accelerate toward it while your head remained in one place, and you'd be stretched out over an infinite time frame. Or I'd have to ask my niece, who's an astrophysicist. But.
All right. Wow.
By the way, if you go out at...
it would take you a while to get there, though.
I understand.
If you go out in the just pre-dawn hours now for the past several days, and I think for the next several days, there is an unusual alignment of the planets. You need your, I need my little Star Walk app to figure this out, but it was pointed out to me by one of the rowing coaches who's an amateur astronomer, where you can see all lined up Venus is the brightest one, and Jupiter and Mercury. Apparently, the ability to see Mercury with the naked eye is pretty rare. And this morning they were lined up just beautiful, all in a line, going from, Venus shows up really bright, and then Jupiter down at an angle from that, and then right on the horizon, Mercury sitting there with a little dot. So go check it out, maybe tomorrow morning.
Neat.
Cool.
Speaking of early morning hours, Rich, what do you have for us?
So this is a pick that was sent to me by Grant McFadden, which is a only somewhat dated 2012 lecture by a University of Central Florida physicist named Dan Britt that was sponsored by the Florida Institute of Human and Machine Cognition, titled Orbits and Ice Ages, A History of Climate. And it's about an hour long, but worth a watch because it's a really nice summary of the whole climate change thing, and useful if you ever get into conversations with people who are spouting the climate denial tripe of things like, well, it's colder now than it has been. It's been warmer than this before. Climate change is cyclical, blah, blah, blah. And it's interesting because he starts this lecture by relating all of those arguments and saying, this is all true. But it is also true.
That's right.
That humans are warming up the climate now, and then gives a lecture that goes through all of that. And it's really very, very nicely done. I appreciated it.
Nice.
So worth a look.
I've watched a little of it so far, and he's really entertaining, too.
He is entertaining, yes.
He is the Pegasus professor. What a great
Ooh.
Share.
Yeah.
The flying horse professor.
Oh, wow.
Nice.
Does he actually get one?
Nobody gets a lifetime of mobile oil.
It's nice because he goes through also the methods. He goes through the science of how we know all this stuff.
Right.
And then at the very end, some denier in the audience stands up and says, "Where's the data? Where's the data?" And he had a beautiful opportunity to smash this guy, but he kind of blew it. He kind of shrugged him off. But it's a good example of how a denier works as well.
Yes,
because he basically has a couple of words ending with "I don't believe it." Well, sorry, I can't do much for you.
It's true whether you believe it or not.
Yeah,
that's the idea.
Mm-hmm. Dickson, do you have a pick?
I do. It's written down below, though. You'll have to transpose it up to where my name is. It's
all the way down.
I know that, but I put it up there. Somehow it got down here. It's the 2018 Best Pictures of the Year for the National Geographic. Stunning. Absolutely remarkable stuff. Some of it not all good. I mean, it's not all pleasant animal pictures and stuff, but these were this is selected from, I think, about 100,000 different photographs.
Including yours?
No, mine didn't even make the cuts. No, they were okay photographs, but they're nothing compared to what these people can do.
100 images, right?
100
Wow.
images. And they're
There's some cool ones
just
here,
remarkable. Yeah. The one that won the whole nature contest last year was this orangutan that was waist deep in water and hiding behind a tree and sort of peeking out saying, "What are you doing here? This is my place." And that was
that's very articulate.
Yes, he was a very educated orangutan. He was fairly old, had been to graduate school, that sort of thing. So I love visual imagery anyway, so I selected
The
that.
seagull one.
These are great.
Cool. Seagull is cool.
Hope you all enjoy them.
These are really good.
These are awesome.
Yeah.
Yeah, I'm going to just sit here and look. I don't know about this Elvis guy, Well, that's not a big deal, but okay. Right. Okay. We shouldn't be looking at these now. We have picks. Kathy, what do you have for us?
I picked vaccinefinder.org. The way this came about is a friend of mine who had the first shingles vaccine had been trying to get Shingrix and hadn't, and instead got shingles. Pretty bad case of it. It sounds like very painful. And so I had the first of the two Shingrix vaccines, and it took a long time to get that. And there's a shortage, and there's no explanation for why there's a shortage, but it's just a well-known shortage. And if you go to this vaccine finder, it can point you to basically for all vaccines what different drugstores have available, and then for any of them they say call to make sure that they have them there. And so today I called 4 and got my name on the waiting list for 4, and I'm already on the waiting list at the university. But the idea was if you went far enough out into more rural areas, you might find places that worked for this friend of mine's friends who were able to get it. And now that she's had shingles, you have to wait until it completely clears up before you can get the shingles vaccine.
So you said that she had the original vaccine and got shingles?
Yeah.
Bummer.
Yeah.
That's not good.
Yeah.
So I just took this little survey. I need flu, which I got this year. Tdap, I just had a Tdap booster last week. 10 years. Zoster, I had Zoster the first year we started TWiV, and actually I talked about it on on the 8th episode. Should I get a vaccine still?
Well, so you've had the original shingles vaccine, and yes, you should go ahead and get the Shingrix.
I didn't get any shingles, but I had Zoster.
You said the Zoster. Well, you mean you got the disease.
I got the disease.
Yeah, you should still have the shingles vaccine, and you should get Shingrix.
And then pneumococcal. Yeah,
you should have that at 65. You should start with 13 and then get the 23.
Okay, and I'm going to be 66 next week, so I'll go and do this.
Yeah.
All right.
You can find out all your local drugstores that.
I was just at my doc. He should have given it to me.
Yeah, that is weird.
All right. Well, I have that's pretty cool. Thank you. Yeah. My pick is a interesting website of a person who makes science gifts, like crabs and hearts and other organs. And there's some viruses in there too, some insects. Here's a cuttlefish, micro.
The keychain that you got.
The herpes?
Blossom Domania gave you.
Oh, it's from here?
Yeah.
It looks like it is. Let's search for herpes. Chris Taylor is the artist here who makes this stuff. Goodbye, Dickson. You coming back?
Coming back.
Yeah,
these are all jewelry type things, right?
Yeah,
they look like
the jewelry type
keychains. Yeah.
And they're in different, like animals, plants, anatomy, cell biology, earth science, astronomy, music.
There's a keychain that's an antibody with a virus.
Yes.
I think I have
spinal column keychain is pretty creepy.
You can get a bacteriophage thumb drive.
Oh, that's cool.
That'll give your computer a virus.
I have the same. Yeah, Blossom gave me the antibody with virus. So I like the beetle earrings. Those are cool. If I wore earrings, I would wear those. Would you wear beetle earrings, Kathy?
I don't have pierced ears, so it's not an option.
If you did, would you wear them?
Maybe. I don't know.
You can wear earrings, Vincent.
What did you say? I can wear earrings?
Yeah, why not?
I don't really want to, no. Yeah, it's a school site, so check it out. Taylor Custom. And I just stumbled on it recently, so it's cool. And we have a listener pick from Anthony, who gives us a New York Times article. The Yoda of Silicon Valley, Donald Knuth, master of algorithms, reflects on 50 years of his opus in progress, The Art of Computer Programming. And Anthony quotes, in issue 33, Mad Magazine published a partial table of the Pochzebi system of weights and measures developed by 19-year-old Donald E. Knuth, later a famed computer scientist. According to Knuth, the basis of this new revolutionary system is the Pochzebi. I don't know how to say that. Does anyone? Pochzebi, which equals the thickness of MAD issue 26 or 2.26, etc. millimeters.
Actually, that quote I got from, I don't know, something where I was following up on this guy. That was his at the age of 19, this future computer science whiz. That was his first publication, basically.
Yeah.
And I think that was even published in MAD or something. And if you look into that system of weights and measures, it gets more and more bizarre.
It's a really interesting article, though. It's a good story. Nice. Good read.
He is a notorious perfectionist and has offered to pay a reward to anyone who finds a mistake in any of his books. Wow. You know, that's a good way to find mistakes.
I'd go broke.
Frankly.
And Bill Gates says something about if you can get through one of these volumes completely, send me your resume.
Yes.
I found that if you have a textbook, your students will find the mistakes. They like to do that.
Always. Trust me, this is absolutely true.
Well, that is TWiV 527. It is the last TWiV of the year. We hoped you enjoyed this year. It was a momentous year, which we'll summarize next week. We'll do our year-end summary. It's a lot of traveling, and we had the 500th episode and so forth, and we hope you liked it and look forward to more next year. Which, of course, if you really like it, you could support us financially. Go to microbe.tv/contribute for different ways that you can do that.
You could support us financially even if you don't like it.
Correct.
Yes, you could. You know, the funny thing is I saw some of the results of the poll on TWIM, you know, this asm.org/TWIM poll. And there's one question. Do you like, dislike, neutral, not like the hosts of the show? It's one of the questions. One person didn't like the hosts.
Really?
Oh. I'm like, why are you listening if you don't
exactly right?
They like the parasites.
Maybe you could support us even if you don't like us. That was TWIM, yeah.
Not TWIP.
Okay. There's nobody. None of us are unlikable.
No, that's exactly right.
But I guess to someone somewhere, someone can be unlikable. So anyway, yes, microbe.tv/contribute. Questions and comments, TWiV@microbe.tv. And please subscribe. It really helps us when you subscribe. We get numbers. We can tell how many downloads there are for every episode. It's really good for us. And please do that. And you can do that on your app that you use to listen to the podcast. Dickson Despommier can be found at many properties on the web.
Thanks to you.
TheLivingRiver.org, ParasitesWithoutBorders.com. Despommier.photoart.com is pretty cool.
Thank you.
I built that one too.
You did? You built all of them.
You put your photos there. They're really nice.
Yeah, thank you.
And so thank you, Dickson, for the year's worth of podcasting.
This was very fulfilling. My capsid is full.
You missed us a few episodes. Right?
I did. I was in Portugal last week and the week before that, but I highly recommend that, by the way, in case anybody has never been to Portugal. Just go. It's an amazing place.
I've never been.
I should, if you like food and good alcoholic beverages of various sorts, but mostly wines and ports, none people are nice to.
Well, we have at least one listener at Persao University, right? Fernando Persao.
Yeah.
Something like that. He writes periodically.
Exactly.
So Dickson, you didn't see him there, right there.
I was looking for him the whole time, and I didn't see him, not once.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
And thank you for the year's podcasting.
Absolutely.
Oh, exactly.
Kathy, how did your concerts go?
Great.
Good.
They were a lot of fun. Yeah.
Good.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
And, of course, thank you for the year as well.
Indeed.
Yeah, absolutely. I enjoy this more and more. It's good.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan, for today and for the year as well.
Exactly.
It's always a pleasure, and it has been all year. Thanks for having me.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Can you do a crow now?
Sure.
It doesn't sound like
No, it sounds like that didn't sound. Thank you.
Kathy's. Everybody's crowing.
Not me. I'm staying out of this.